So we left off talking about mitosis and the cell cycle, and today we're going to start talking about meiosis. Now, the first thing you're going to notice is, of course, that meiosis sounds an awful lot like mitosis, and they're often confused, and so it's going to be your job to figure out the differences between the two, and we're going to do like a comparison and contrasting chart between the two, because there are some similarities, but ultimately they're extremely different for what we need to know. So when you start reading about this in your textbook, you're going to start seeing a lot of terminology that looks pretty simple. You can see that I have here a 2n leading to a 2n, whatever that means, and then I have two 1n's. Well, you know that 1n plus 1n equals 2n, so that might make some sense. But then down here I have 1n leading to two 1n's. It's really confusing, and it's a lot. And so this is like the most simplified diagram that you're going to see from the textbook. And this honestly is just really, really dumbing it down. And it's just so that you can focus on one thing, and that is what these ends are. So you probably remember from regular biology years ago that humans have 46 chromosomes. Okay. And what that actually means is not that we have 46 unique chromosomes. That's not what we have at all. We actually have 23 pairs. So they're all paired up next to each other. So if this is chromosome 1, that's also chromosome 1 because we have two copies of each of them. So that's what your book means when it talks about this term diploid. Okay? It basically means that I have two copies of chromosome 1. I have two copies of chromosome 2. Maybe it looks a little shorter. So here's 2. I have two of those. And I have two copies of chromosome 3. Maybe they look a little longer. Okay. And then I keep going and going and going until eventually I have 46 total chromosomes. So if this is a chromosome right here, and this is a chromosome, both of these are chromosomes, then there's two. So far I've drawn 6, and I need to get all the way up to 46. So basic math will tell you that you need 23 pairs of chromosomes in order to reach that 46 that make up humans. So within your body, within your cells, within their nuclei, you will find 46 little chromosomes. And we always draw them paired up together by size. So you'll notice that like a big one's always next to a big one. And then maybe there's a pair of small ones, etc. And in reality, they all look very different from this. And you'll rarely get them in a straight line like this. We're going to look at some karyotypes to really show what they look like in reality. But this is just a simplification, of course. So So if I were to draw 46 of these lines, that would be a diploid human. There are 46 chromosomes. But what you'll notice is that we have this 2n thing. So n is basically the number of chromosomes. So if you have 2n, then that means that you have 2 times the number of chromosomes. So what humans actually have is 23 chromosomes. Okay. But we have 2 of each. So that's what it means to be diploid. You have 2 of each chromosome. Okay? And so that's how we represent 2n. Okay? So 2n and diploid, those are saying the same thing. If you're just 1n or n, we call that being haploid. That means you have one copy of every chromosome. And that's going to become super, super important when we go through meiosis. I know it's a lot right now, and it's mainly just terminology at this point. We don't even know what these mean. Like, what is meiosis 1? What is meiosis 2? We're going to get there. So here is kind of the next level up of what I would expect you to be able to see in a meiosis diagram. So with meiosis, you're always making gametes. Okay, so you're making sperm and eggs. Hopefully you remember the term gametes from regular biology or possibly from middle school biology. If you do not, gametes are simply just sex cells. They're sperm and they're egg. Those are easy enough to remember. So gametes, again, sperm or egg, are pretty unique. So they are haploid. Okay? So gametes are for humans and any other sexually reproducing organism that we're going to worry about. They are going to be haploid. That basically just means that they have one copy of every chromosome. Okay, so what we have to do is we have to take a parent cell So like this right here. So this is a parent cell. So parent. I have to take that and I have to turn it into a haploid cell. So this is diploid. Okay? Now in this simplified diagram, what we have here are only two different chromosomes. Okay? You'll notice that there are two different sizes. I have this large one, so this is 1. Let's just call it 1A. Here's the other large one. Let's just call that 1B. You'll notice that they're different colors. That's going to become important in a bit. Here we have 2 and another 2. And it doesn't really matter which one you give A, which one you give B. I'm going to give the blue one A so that it's just consistent with chromosome 1. It really doesn't matter. So the best thing to do, because this is kind of an overwhelming amount of information all at once, is just break down each individual step. Just try to figure out what's happening at each arrow. Okay? So from this first cell over to the second cell, you may notice that I went from just like having a regular 1A strip to now having like a 1A, but it's got a friend kind of attached to it. And if you'll recall from the cell cycle chapter, this is when the DNA has copied itself. It has replicated. Okay, so I'm going to put right here in that arrow, this is replication. And it's really hard to write with this mouse, so bear with me here. This is replication. So I copied the DNA. So if you look closely, you'll just notice that every single one of these chromosomes that are all bundled up here has now gotten a partner. That's it. They've just gotten a partner. And what you also might notice is that the large ones like to hang out near the other large ones, and the small ones like to hang out near the other small ones. And that's going to become really, really important because you will see that this is chromosome 1, that's chromosome 1, this down here is chromosome 2, and this is chromosome 2. Remember, we had A's and B's and all that, and that's really important. But for now, let's just look at the colors, because the color kind of denoted that as well. So for now, let's just think about blue as being A and purple as being B. So, excuse me. What I have here in this bubble is I had 2 X's that were blue, different sizes, and 2 X's that were purple of different sizes. Now those colors are just arbitrary. That's just something that we have done as scientists to basically simplify things and help our brains to visualize them. Okay, they're not actually blue and purple. All right, but when I go from this step to this step, you may notice, so what's happening in this arrow, you may notice that now this one that was all blue, it's got a little bit of purple. And this one that was all purple now has a little bit of blue. And the same thing happened down there. So that's really, really important. And we're going to be breaking down that process. This is what we call crossing over. So if you'll remember, DNA is information, right? It's basically what codes you to become you. So it's pretty important what is on that information. So what we see happening here in crossing over is they're actually changing their information. They're exchanging it. So we see that this blue X and this purple X are exchanging little bits. You'll notice that like if I were to cut those, like cut there and cut there, I could like switch them back, right? They're the same size. They are in the same spot. But it's really, really important that you notice that they're exchanging some information here. And we're going to come back to that later. But then we need to go from this cell to 2 cells. We need to figure out what's happening in these arrows. Okay, so basically, you'll notice that now I only have chromosome 1 and chromosome 2. Well, before I had 2 copies. Over here I had this one for chromosome 1 and this one, right? But now I've only got 1 of chromosome 1. And how many of 2? Well, I only have 2. Now you'll notice that there are 2 little strands that make it up, but anytime they're still together, they're still connected, we still consider that its own full chromosome. So that's still all just one big chromosome. Okay? And down here, I only have chromosome 1 and chromosome 2. Okay? So basically, you have to ask yourself, how many copies of every chromosome do I have in this cell, for instance? Okay? In that cell, how many copies? Well, chromosome 1, I've got that X. Chromosome 2, I've got that X. So I have 1 copy. And if you'll recall, when it's 1 copy, we call that being haploid. So until this step, we have been diploid this whole time. And now we're haploid. That means we have one copy. And then these cells then go on to split, and they make sperm and egg. Okay? And you'll notice here that the sperm and egg are also haploid. Okay? And they kind of get rid of that extra little bit. So we saw here that they had replicated, and so they have like a sister strand that's still attached to them. We're going to break down what that means because that is important, but they separate in this step. And so then your sperm and egg have just one copy of each chromosome, one copy of chromosome 1, one copy of chromosome 2, and they're not replicated. It's just a single strand. So it looks like I took half of this original parent, roughly. Okay. So the same thing happens down here with ova or egg production. So up here with sperm production, it doesn't matter at this point, the differences, when you're looking at a diagram as simple as this, because really the only differences that we care about for now are going to be sex differences, so the X and Y chromosomes. So just bear that in mind for now. Now with eggs, you'll notice that with sperm we made 4 sperm. And we do the same thing with eggs, but one of them is a lot bigger than the others. And that's just because eggs have to have a lot of stuff in them. And so most of these, in fact, all of these are not going to work. They're just going to get broken down later. So they just kind of get made only to be broken down because the cell doesn't have a better way of making just one. Okay, so they make one egg that combines with any of these sperm, and that occurs during a process called fertilization. And when you fertilize, you form the baby, which when it's just a single cell, there's only a single cell here, it's called a zygote. So my question to you to think about right now is what ploidy level does this zygote have? Is it haploid or is it diploid? Is there one copy of every chromosome or is there 2 copies of every chromosome? Okay, so take a quick second to think about it. So you'll notice that I have a big chromosome and another big chromosome, a small chromosome, and another small chromosome. So I have 2 copies of chromosome 1 and 2 copies of chromosome 2. Now they look a little different, they have different colors, but that's totally fine. The information there, it still has the same genes. It might have different alleles, but it has the same genes. So this individual is now diploid. So what I did is I took one haploid sperm, one haploid egg, combined them, 1n plus 1n equals 2n. Okay? Now, this is kind of meant to break us down into the subsequent steps that were just kind of implied in that previous diagram. So this is just a reminder that chromosomes do replicate, they copy themselves, and this occurs during the S phase of the cell cycle. And when you do that, you now have 2 chromatids, 2 sister, meaning identical, chromatids that are attached at a centromere. So here, when I have just a single strand right here, and then a single strand right here, they're called homologous. That means basically this is chromosome 1, and that's chromosome 1, but they're not attached. Okay, it's just you have 2 copies, right? Chromosome 1 is going to copy itself. And so that means that now, instead of having just those 2 chromatids, 2 chromosomes, I now have 2 larger chromosomes made up of 2 sister chromatids. Okay? So this part right here is very important. This left strand is now a chromatid. This right strand is now a chromatid. But together, they are sister chromatids because they're identical. And when they're still lumped together like that, anytime they can't separate, they're not separate yet, we still consider it just one chromosome. Even though it has 2 chromatids and it looks very different from what we called a chromosome before, because it's twice as much, it's still considered a chromosome. And that's a really confusing thing, but it's just arbitrary and you just need to accept it. So a chromosome is anything that is self-contained. So if it's by itself, it would be considered a chromosome in the context of meiosis. Okay. So this is just a reminder about what it is that we're talking about here. We're talking about DNA, and DNA is this double helix that you guys know very well. And DNA likes to condense inside of nuclei just because, like, if it's floating around like that, it's insanely long. We don't want to get in places that we don't want to. We don't want it to try to interact with anything. We like it in a nice condensed form whenever we're trying to move it so that we can kind of keep it out of the way. So what it does is it basically just wraps around some histones, some proteins, and it kind of looks like beads along a string when you take an electron micrograph of it. And so this is basically just a DNA strand wrapped up around a bunch of beads, and millions of times later, you get a chromosome. So that's really, this is what we see right here anytime we're working with electron microscopes or anything like that. Here I have one sister chromatid. Here I have another sister chromatid. And together, you'll notice that they're still attached. They're attached at the centromere. That makes up a single chromosome. So just a little bit of practice for all of that terminology that we unfortunately have to know. So meiosis is a little different, and I have a spelling error up there, but that's all right. Meiosis is a little different from mitosis because it's almost like it's mitosis happening twice. Okay? The stages are very similar. So if you remember the stages of mitosis, like we had prophase, we had metaphase, etc., we're going to see those again, and there's going to be a lot of similarities. So it's not really learning a new process from scratch. It's really just learning an old process twice. Okay, obviously there are a few differences, but it's really not too bad. So just a little bit of a throwback to the previous chapter. Interphase is the phase that most cells spend most of their time in. So it's basically when they're just doing their job. They have two copies of every chromosome. They have a 1A, they have a 1B, they have a 2A, they have a 2B. Okay, two copies. So this is diploid. Okay. Then they're going to replicate. Okay, that happens during S phase of the cell cycle. So they're going to replicate. And then we are going to now have some larger chromosomes. They're still chromosomes. So this whole thing right here is still 1B. This whole thing right here is still 1A. This is 2A, etc. Even though it's made up of two sister chromatids, the whole thing, it's combined, it's one chromosome. Okay? So they're still homologous. So here we had a homologous chromosome pair for 1A and 1B because they're both chromosome 1. Okay, so they were homologous. Homologous. I'm just going to write homolog because it looks terrible. And then over here, they're still considered homologous. So I still have 1B and 1A next to each other, and they are still homologous, even though we have 4 chromatids now. Okay? And we're basically going to split it and then split it again. And you'll notice that we did some of that color exchanging again right here. Remember, that was called crossing over. We're going to explore that a little bit more. But this is kind of just to put into context all of these terminology, these terms rather, that we need to know, like homologous chromosome, sister chromatid, what a chromosome is, because that changes. So as we start to go through this process of meiosis, you'll notice that there's a meiosis 1 and there's a meiosis 2, and each of those has similarities to mitosis. So we're going to see the same terminology used for the phases of meiosis as we did for the phases of mitosis. So we have prophase 1. I remember that was the first stage of mitosis. It's also going to be the first stage of meiosis. So in this stage, our chromosomes start to condense, so they get all coiled up. They start to form that shape that you're more familiar with, all right? And then we can see it under a microscope. The nucleolus, right, so the thing that is protecting the chromosomes, starts to break down, and spindle fibers start to form. So those are the things that'll like eventually grab the chromosomes and move them around. They're starting to form, okay, from the centrioles. And then homologous pairs are going to pair up. Or excuse me, homologous chromosomes are going to pair up. So what that basically means is chromosome 1 and chromosome 1 are going to hang out near each other. So here's 1A, here's 1B, whatever. Okay? And they're going to get next to each other so that they can do this little exchange. And so what you see here is basically the exchange has just happened. Okay, you'll notice that there was a dark blue chromosome and there was a light blue chromosome, but now one of the chromatids of the light blue is still normal. One of them is mostly light blue with a little bit of dark blue. And then the same is true of the dark blue. There's a fully dark blue and there's a dark blue with just a little bit of light blue. Okay, so that's that crossing over, and that's going to become important later. So after prophase, we have metaphase. Meta, just remember that M means middle. Okay, same thing for mitosis. Basically, these spindle fibers are going to start moving our chromosomes to line up at the middle. Okay, we have like a metaphase plate again. Okay, they're going to line up at the middle of the cell. That's why we think of it as the middle for M. And they're going to basically orient themselves at random. So they need to pair up. So they need to have chromosome 1A and chromosome 1B. They need to be next to each other. That part needs to happen because they're still next to each other from when they crossed over. Okay? But it doesn't matter which one's on top, which one's on bottom. It's basically whoever gets there first. Okay? And there are chemical signals that are doing all this. Obviously, the cell is not sentient. It's not thinking about this. Just like in mitosis, we see anaphase where they start to split. So we had a chromosome here, and it was made up of two sister chromatids. After anaphase, I have split those sister chromatids. And so now I actually have, there's a chromosome, there's a chromosome, here's a chromosome, here's a chromosome. Here's a chromosome, here's a chromosome, here's a chromosome, here's a chromosome. Basically, as soon as they split, they become their own thing. And so now we call the self-contained chromosome what it is. Okay, so that chromatid, or what was a chromatid, is now a chromosome. Okay. Then we have telophase, so that's where they start to move to either side of the cell. Telo means far, so they're moving far away. Okay, and then we start to see the nuclear envelopes reforming on each of the what will become a cell. Okay? And there's a cleavage furrow. Remember, that's just basically like you're twisting the cell right here. You're like cinching it, and eventually you'll separate it into two cells. Okay? Now, these cells are haploid because here, you know what? My brain totally just messed up. For some reason, I thought that we had already split our sister chromatids, but we have not. We have only moved them. So we are still at 4 chromosomes total. Here's chromosome 1A. Here's chromosome 1B. Here's chromosome 2B. Here's chromosome 2A. Sorry, I got ahead of myself. I really like meiosis 2 more because it's more interesting. But here we have 2 haploid cells. Okay, and so we still have just 1 copy of chromosome 1 here and 1 copy of chromosome 2 in this top cell. Okay, then we're going to basically do it all again. And so this is what I was just picturing, and my brain just totally blanked because I'm on vacation too. So during prophase 2, we have to break down that nuclear membrane that we just started building up. And I know it kind of sounds silly to build it up and then just break it down immediately, but our cells don't really know any other way. They can't think about how inefficient that is. Okay, they just do things. So our centrioles are going to move to either side again. So those are little cylindrical shaped things. They're going to move to either side of the cell. The spindle fibers are going to attach again. And this time, they're going to actually separate the sister chromatids. Okay? And so now we have a chromosome, a chromosome, a chromosome, and a chromosome. So up here in this top diagram, we have 4 chromosomes, and there's going to be, excuse me, 2, 1, and 2 in the top cell, and 2, excuse me, in the bottom cell. And then the same is true down here. Okay, so the reason that I'm just combining, excuse me, metaphase and anaphase together is because it's really the same thing as what we saw before, except we're doing it again. Now that we're in meiosis 2, we put a 2 after everything. So instead of it being metaphase 1, we're doing metaphase 2. Instead of it being anaphase 1, we're doing anaphase 2. And you do need to know the differences between those, and we're going to practice it a ton. Okay, and I apologize for the little confusion there. My brain just is not working yet, and that's all right. But we will once again end with telophase. And with telophase, we are going to end up with 4 haploids, so that's the N, gametes, so 4 sperm or 4 egg. Now, remember with eggs, really only one of them works, right? So like these 3 don't work, but that one does, or something like that. Okay? And that's just because they need a lot of resources. Okay? Here's an overall diagram showing the entire process. So we start an interphase. This is what most cells are doing all the time. Okay, they're just doing their job. And then during interphase, they replicate their DNA, they copy their DNA. And then we can enter prophase. So during prophase, you'll notice that the nuclear membrane starts to break down, so that stuff can kind of move around a little. In metaphase, the chromosomes start to line up. Okay? And this diagram does not have crossing over at this point, but if you want to just picture crossing over, you would just exchange a little green and a little purple, and then in every subsequent picture, this green would have a little bit of purple, that purple would have a little bit of green, and then the same thing for the chromosome 2. Okay? They're going to split, and that's going to form two distinct cells. So here we have two distinct cells. We're still haploid. Okay? So we are haploid now. Okay? As soon as those things split and form two unique cells, we're haploid because we have one copy of chromosome 1 and one copy of chromosome 2. Okay? Excuse me. Then we're going to start going through what we call interkinesis, which is basically just, if you know what inter means, it means between. Kinesis just means movement. And basically, we're going to see a step that is intermediate between meiosis 1 and meiosis 2. Some cells might spend a little bit more time just kind of chilling between meiosis 1 and 2 if they need a little bit more energy, or some cells will just jump straight into meiosis 2. And so we're going to go from prophase 2 to metaphase 2. So now we have our sister chromatids that are forming one chromosome lining up along the metaphase plate. Okay. In metaphase 2, they're going to separate. So now at this point, I have a chromosome, a chromosome, a chromosome, and a chromosome. So 4 chromosomes in that bottom cell, but eventually it's going to be 2 in the left cell and 2 in the right cell. And the same thing is happening up top. So these things are really easy to picture as these like little X-shaped things, but remember there's a ton of information on there. And so these things change a lot throughout meiosis 1 and meiosis 2. And so it's really important that you start to picture what it is that they're doing. So in the top part of this little diagram, I have meiosis 1. Okay? And in meiosis 1, you have your homologous pairs. So I have chromosome 1, chromosome 1. They're different colors because you got one from mom and one from dad when you were born. Okay. And they're going to be held together. And basically, that is going to be what allows them. You don't need to worry about all these different terms like microtubules and stuff. We've seen those before. You don't need to know it. You don't need to know the mechanism. You just need to know, that they exchange some information. So there's a little bit of red on my blue one now, and a little bit of blue on my red one. Okay. So now that we have a little bit of more variety between my two, so instead of having all blue, I have an all blue sister chromatid with a mostly blue sister chromatid, an all red sister chromatid with a mostly red sister chromatid. Okay. We're going to jump into meiosis 2. Okay. And remember that sometimes we lump together parts of prophase and metaphase into pro-metaphase, so your book will talk about that. You don't need to know that difference or anything like that. Okay. But basically, we're going to separate these sister chromatids right down the middle. Okay. And that is going to split them into 2 distinct chromosomes. So now in this image, I have a chromosome here and a chromosome here. So we went from 1 chromosome to 2 chromosomes in meiosis 2. Okay. This is just a different diagram of that crossing over. So the sister chromosome, homologous chromosomes line up next to each other so that they can exchange a little information. And so that's why we see like little mismatches. We see like, oh, I have yellow and green. I have green and yellow or whatever. Okay. And this is really important because anytime you have diverse, because this is what will become a chromosome. This is what will become a chromosome. This is what will become a chromosome. And this is what will become a chromosome. Right now, we have 2 chromosomes. We have a sister chromosome here, and a chromosome made up of 2 sister chromatids here, and a chromosome made up of 2 sister chromatids here. Eventually, after meiosis 2, you're going to have 4 chromosomes, and those are the things that I just put big old ovals around. Okay? And the importance of that is really hard for me to boil down. So we're going to do an activity in which you guys basically get to see why it might be useful or why it might help a population to have this crossing over happen. What would happen if you don't do that? And so that basically allows for more variety, so more different types of people or individuals if we're not talking about people. But another thing that allows for different combinations of chromosomes in our gametes, in our sex cells, is that the actual chromosomes themselves move around. And during metaphase, they line up, but there's no like absolute way they're going to line up other than they're going to line up with something of the right size. So like they're matching chromosome, but it doesn't matter who gets on what side. And so we're going to explore this some more as well. But basically, there, even without crossing over, even if we didn't exchange any information, there is 2 to the 23rd different possibilities of chromosomes that you could have in your gametes, in your sex cells, which is something like 8,400,000 different possibilities. Okay. So, or perhaps it's 8 billion. I don't remember. Bad at math in my head now. So, excuse me. It's basically infinite. So like if you wanted to have if your parents wanted to have a kid that was exactly like you, because you're like the perfect kid, well, that means that you have to have this, let's say sperm, let's say this is from dad, and this egg. Okay, so that means that there's a 1 out of 2 to the 23rd power of your dad making the exact same sperm, and that being the one that fertilizes the egg. And there's a 1 in 2 out of 23 that your mom makes the exact same egg, and excuse me, and that's the one that gets fertilized by that sperm. And so these two things have to happen in order to make the exact same individual. And so it's like a 1 out of 65 billion or some crazy number percent chance, or trillion, I don't even know. So it's very, very, very unlikely that 2 individuals are going to have the same DNA from their parents. It's technically possible, but it is so unlikely, especially because we haven't even accounted for crossing over in these calculations. So there's even more diversity, and that's just really, really important. But if I were to boil down why meiosis matters, it basically allows me to create a unique haploid sperm, a unique haploid egg, and combine them via fertilization. So that's our process here, fertilization, to form a diploid zygote. And this baby is unique. It's not exactly like mom. It's got half of mom's DNA. It's not exactly like dad. It's got half of dad's. But it's got a unique combination. So they're going to be their own unique person. Okay? If you are super confused, because this is a really, really confusing topic and it's filled with diagrams, and you honestly just kind of need to sit down and diagram it yourself, there is a decent crash course video on it. So here's the URL if you want it. Obviously, you can just YouTube crash course meiosis. It's very, very useful, but what I will say is don't rely on it. This is an instance where you really, really, really want to diagram the process yourself. If you do not do that, if you don't spend the time to do that, you will not succeed in this chapter. I will tell you that now. So with that, good luck, and I'll see you guys for our next lecture.